And I think we are now recording. So, hello, I'm Paul Humphreys, and this is Rheophilia, which is a rather strange little name, but it actually means flow-loving. Interviews in freshwater science. And today I've got Dr. Terry Hillman, AM, which means you're an member of the Order of Australia, which I think you got in 2014, Terry, is that right? Sometime around then.
I don't know, yeah.
For services, I was actually looking this up because for services to science and freshwater management and things, but I also looked up your name just to see if I could find some old photographs of you, just so I could embarrass you, but I couldn't find any. But I did find that you actually co-authored The Complete Idiot's Guide to Surviving Divorce. Did you know that?
Oh, that's an American guy, isn't it? He's
oh, it's not you.
No. He's a divorce counsellor.
So I think it's always a good thing to realise that you've got a person with the same name who does interesting things that is quite outside your field. I think I've got a serial killer or something. I think there's a Paul Humphreys who's a serial killer, and also I think certainly somebody who played for in the band. I think it's a British band, Orchestral Manoeuvres in the Dark, OMD. Yeah. So there you go. Anyway, Terry, thanks for agreeing to be my third guinea pig interviewer here, and the oldest living member of the trilogy, the triumvirate, whatever the term is. So you've got a wealth of history, and I'm looking through your sort of what you've been doing in the last 50 odd years or something, and it's quite substantial. I think I want to sort of start off by, if that's okay, asking you, because you've had a fairly checkered history, and I've got he sort of started off by doing a diploma in agriculture at Dookie College along the Broken River, where I've actually spent a small amount of time myself. What got you interested in the natural world, and clearly with some interest in agriculture to begin with? What's your background? Where did you come from?
I was born in Albury, but just at the beginning of the war, Second World War, not
the First World War.
Crimea,
the Boer War.
And then my father at that stage was a school teacher. He later had to give that up for health reasons and became a farm hand. I'd always liked living creatures, particularly insects. I still do. And so, but at the time, we obviously didn't have any money, and I went to a school where you only went to grade 8 or something, which happened to coincide with the time you could get out of school and go and get a job in a factory somewhere.
So what was the school you went to in Albury?
No, this was in Mildura.
Oh, Mildura.
Yeah.
Okay.
So, sorry, I left that bit out. So that was fine, and I saved up a bit of money and bludged a bit more from my old man. And sort of the thing that, at that stage we were living only about a kilometre away from CSIRO's Division of Horticulture research station, and that looked to me pretty interesting. And
when was CSIRO first set up, though? What?
Oh, yonks before that.
Yeah, okay. But was it CSIRO or was it SIRO?
By this time it was CSIRO.
Right. Yep.
And I think from memory, Bob Menzies might have set it up financially, but I can't remember for sure. I wasn't there.
Yep.
And anyway, so that looked like something I could do without having too many qualifications, and still get to do an interesting job. So I went and did that. I'd spent a couple of years, I think, working as a plumber's helper. which is a code word for digging holes, basically.
Oh, it has a water component to it, though, doesn't
It does have a water component to it.
Actually, I think I remember though. You still retain some of the skills. I still have a vague memory that when I was working at the MDFRC, Murray-Darling Freshwater Research Centre,
Yeah.
that you offered to fix taps or do something because you'd had still retained some skills in that area. Is that true?
Okay. Oh, you better not let it out that I fixed taps on the on the mains.
Oh, it wouldn't be allowed nowadays, would it?
No, it would not. Anyway, and so I did all that, and I got a job in the Division of Entomology and CSIRO, which was a technician's job, which was
Right. Yeah.
really good, and I was lucky to fall in with the team who was doing applied work, population ecology work.
Were there any big names at that time doing that?
Not that well. The division division main offices where I was working in Canberra. Nicholson, AJ Nicholson, was the head of the place.
Mm-hmm.
He, as you know, was a big mover in population ecology. So it was the start of a lifetime of being lucky enough to be in one spot where things were going on and pick up some tricks.
So you were only in your early 20s when you started there.
Yep.
So you didn't have any formal qualifications at that stage, apart from the diploma of agriculture.
Yeah.
Okay.
That's right. But CSIRO at that stage had a fairly generous arrangement whereby you could study part-time, get about half a day a week off. And so as I was in Canberra, in fact, across the road from ANU, it was a sensible thing to do.
But then you went on and did I gather, from 1970 to 74, is that at ANU, was that your PhD? Or was that your...
Yeah, that was my PhD.
And on ecology of algal blooms?
Yes.
Why, for a person who was in love with insects, did you go and work on algal blooms?
Primarily because, and this sounds a bit sort of uppity, but primarily because the head of the School of Biological Sciences, RSBS, whose name was Ralph Slatcher, wrote and asked why I hadn't applied for a scholarship in his thing. So I did.
Right.
And that worked out fine. He was an amazing mentor. And also he had a strong policy of supporting and otherwise encouraging overseas visitors to come for short periods and work there. So as a PhD student, you got to talk to and listen to quite amazing people. Slatcher himself, of course, was one of the main people who'd come to understand how photosynthesis worked. So,
can you remember any of the people who came and visited during that time? Or was that going back too far?
That's going back miles too far. I'm not very good on remembering names, and I can't remember any now. But there were a lot, of course, in that more biochemistry area, which didn't interest me directly, other than hearing interesting stories. But during that period, Slatcher at the time was trying to build up a sort of ecology component to his department, and so he appointed Gene Likens, was who I was trying to think of before.
Okay. Yeah.
So he spent a couple of periods there, quite substantial. In fact, he did a lot of his acid rain work from there.
He must have been fairly young at that stage.
He or me?
Both of you.
Yeah. Yes, he must have been, mustn't he? Yeah.
He went on to big things. So you did your PhD on ecology of algal blooms, which was, you obviously presumably found that interesting despite your love of insects. Is it something that has persisted?
I didn't really actually want to do it. There were two projects that Ralph wanted someone to work on. And the other one was the one that interested me, and I think probably Ralph would have preferred me to do it too. But and that was the ecology of seed harvesting ants around the Hay Plain and areas like that.
All right.
And they're quite amazing, of course, because they actually pull all the strings that controls the species makeup of the ant community there. And if you took them out, you would just completely screw that bit of the ecosystem.
So was this PhD the first sort of aquatic-y type work that you'd done up to that time?
Yeah. Although I did a bit of work with a fellow called Alan Weatherly, who you may not
Yes. Sam Lake's
that's
PhD
right.
supervisor.
Yeah, that's right. Yes.
Fish guy.
Yep. A fish et al. Guy. Actually.
Yeah, that's right. Yeah. Was he Canadian? Wasn't he Canadian?
No. He went
to Canada.
He went to Canada with his. He remarried and went to Canada, where his second wife was a Canadian. I think. And in fact, this is all a bit by the by, but as I was finishing my PhD, I contacted him because he'd moved to a town called Tromsø, which is in Norway. I was going to say behind the Iron Curtain. I really meant beyond the line where you get ice frozen.
The Arctic Circle.
Arctic Circle, that's the one.
Right. Similar things, yes, Arctic Circle. I like that. That's good.
Well, you run into one at high speed.
He was sort of above the Iron Curtain, wasn't he?
More or less above it.
Beyond the
Iron Curtain, anyway. So, yeah, and he wrote back and said, yeah, come over, I've got a job for you, which was nice. And I would have had a research vessel and all sorts of nice things. But then one of my daughters got ill. They were also, one of them was also the reason I didn't go do the ant stuff. It would have been camping up in Australia for 6 or 8 weeks at a time doing field work. It wasn't appropriate.
Okay. So you didn't go to Norway?
No, I didn't go to Norway. I got as far as writing and accepting the job application, but didn't get there.
Well, but your PhD, despite it wasn't your favourite thing that you ever did. Interestingly, why was there interest in algal blooms at that stage?
It wasn't directly about algal blooms. Well, algal blooms is part of the issue. It was looking at the sort of minor and sublethal effects of zinc pollution, because Lake Billy Griffin had a zinc problem, and so it was really dull. Cubs honour.
Did you manage to publish anything from that, or wasn't it? Did you lose the will to live after finishing your PhD?
I lost the sort of recognition that I may still be living. It was.
But you went on. I mean, pretty soon after that, after finishing that, you came to Albury to work for the Albury Wodonga Development Corporation, I think.
Yeah.
In an aquatic, primarily an aquatic field.
Yeah. Well, I've left out a step actually because towards the end of my PhD, I can't remember exactly when, Keith Walker, the late Keith Walker, came to ANU on I guess it was a postdoc still. I'm not sure. Maybe it was a.
And he done his PhD at Monash, was that?
Yeah, he did his PhD
with Bill Williams.
Yep.
Yeah.
And then he spent time with another person not called Bill Williams.
Right.
Who lived in Washington.
Okay.
I'll think his name.
That's all right. Before coming to Canberra.
Yeah, before coming to Canberra.
Yeah.
So we've known he accepted that situation before he actually left Australia. So it it was.
Had you met Keith at that stage?
I met him during that process.
Right.
Yeah, and I knew Bill Williams a bit. No, I didn't know him at that stage. That's wrong.
Okay.
But he very generously, because Bill went on sabbatical just about the same time, and he left me access to the key to his office and access to his reprint collection, which was pretty massive.
So this is in those days. It's not that long ago, really. That before computers, before access to enormous databases. That's what we did, didn't we? We printed out. We wrote away to people to ask for reprints of their papers, their scientific papers, and they sent it to in the mail. And they had those little cards that you used to send away to
reprint
them.
requests.
Yeah, reprint requests. It was very exciting waiting for those to come
back. It was, yeah.
It was a slower time, wasn't it?
It certainly was.
Anyway, so you had an access, and I remember Sam Lake's. He had a whole room almost full of reprints. It was, the most important sort of resource you had in many ways. I still have quite a lot of mine. Don't know what I'm going to do with them. But so having access to some of these reprints would have been a fairly big thing.
Well, particularly as I was starting out on a branch of science about which I knew nothing. Mm-hmm. I couldn't tell
so just
from a grasshopper.
Just tell us briefly who Bill Williams was.
Bill Williams was a funny guy in every sense of the word, really. But he came from England to Australia to take up his job as head of the zoology department. I don't think he had a sort of lesser position and work done. And at the time, there was a lot of debate about freshwater science in general, and CSIRO had reaffirmed their view that that wasn't an area that CSIRO needed to worry about. And as a consequence, there was quite a void where we really didn't know anything much about our freshwater systems. And for a while he was building that place up, even then the projects were esoteric at the best and not really seeking to play the main game.
Would you say they were taxonomically based sort of stuff, people describing species and things?
A lot of that. And even now we're sort of well behind the eight ball when it comes to describing the animals that live in those places. Like
Ian Hiscock with mussels and things, those sort of people. It was one of my lectures when I first went to Monash. Yeah.
Oh, okay. Yeah. Ian was rather fun.
Yes, in all sorts of ways.
Yeah.
So Bill brought a different way of looking at things to Australia.
Certainly, yeah. I think he recognised that it was at a very early stage of development and worked accordingly. So he did a great deal with limited resources to build up a source of knowledge.
It would have been an exciting time, though, wouldn't it? Because of the lack of sort of solid sort of ecological research that had been done, especially with invertebrates at that stage.
Yes.
Yeah.
Yeah. Well, non-terrestrial ones anyway.
Yeah.
Yeah. So I don't know where we were now.
Oh, so I just asked you about who Bill Williams was for people who don't.
Oh, okay.
Don't know of him, but you were talking about Keith Walker coming up to Canberra.
That's right. Yes. Yeah. So Keith Walker came up to Canberra, and then Ralph Fletcher was about to go overseas for 6 months or so, and so Keith took up my. We had a committee system of supervision for PhDs, and Keith took over running that.
So he was like a supervisor of your
PhD? Yeah, he was a stand-in supervisor.
Oh, okay. I didn't realise that.
Yeah, which was great because, as you probably remember, Keith, he was fairly fussy about the English language, And I was busily trying to write the stuff up. So
Yes, I think we'll talk. We'll probably. I'll find some way of interviewing or talking more about Keith Walker at some stage. I'm not trying to stop you now because I mean I
okay.
could talk about Keith forever too, but I'm
Yes.
just because he was like Bill Williams, such an important player, an important person in freshwater science in Australia, and a lovely man who.
Yeah.
There's so many nice people in our sort of field. Anyway,
It is. Yes. Yeah. You're right.
and we've lost a few of them in the last few years, which is
Yep.
a real shame. Anyway, it happens. So Keith came up and took over some of that supervision.
Yes.
Of your
he did.
PhD.
Yeah. It was sort of at the writing up stage by then, and
Yeah.
And so it was very, once again, fortuitous that he popped into place at the time when I would need.
And helped you with your writing.
Yes.
Well, I tell you what, he was. I mean, I miss him so much because we wrote edited the Fish book together a few years ago.
That's right, you did. I forgot about that.
And I think for about 2 years, we were communicating more than daily, more frequently than daily about the editing, and he was. I learned so much.
Yeah.
About it, writing and about the editing process, and about being very diplomatic, diplomatic but firm with authors about how to edit 30,000 words down to 15,000 without actually losing any information from that, which he was absolutely a master at, and he was amazing and very patient man.
Yes.
But didn't suffer fools gladly either. He was quite happy to tell people that no, this is the way it's going to be, but he did it in such a nice way.
Yeah.
So I can imagine that he did that with your PhD as well.
He did it in every tiny little crevice in it.
That's right.
Yeah.
A flick of the eyebrow. Or
it took me a couple of weeks to get the hang of how long different sorts of hyphens ought to be.
M dashes and N dashes, all that sort of stuff. Yes.
And as you say, as you demonstrate, they've all got their own little names. And Keith was of a very strong taxonomic bent when it came down to it.
Yes, that's great in when you've got an editor or a supervisor who's helping you writing. I mean, I'm doing the same thing at the moment with PhD students, and I'm sure they get very, very frustrated with me insisting that there's a semicolon there rather than a comma or something. But I think that's it's sort of, you know, in some ways that's, well, not in many, in some ways, in many ways that's our job. A lot of our time we spend editing people's work and writing our own work. The communication of science is such a fundamental thing to what we do,
and pretty well unforgiving as well.
Oh yeah, and it's interesting that it's interesting when you get students coming in who maybe have worked out in the real world, dare I say, for a while, think that somehow science, you know, language of science sort of has to have some level of obscurity or opaqueness to it.
Yeah.
And I keep, you know, I spend most of my time sort of saying, no, it has to be really clear, simple, concise, and that's a real
and unambiguous.
Unambiguous. I always say that your role, your task, is to write a sentence that doesn't need to be read more than once.
Exactly. I say the same thing. I want sentences that, when you get to the end of them, you don't have to go back in the middle and find out what you're talking about.
Yes, exactly.
Complete package.
But we digress. So, so Keith Walker, and was that your beginning of your, you know, working and other relationships with Keith? I mean, did you obviously went on to do quite a lot of work with him?
Yeah, I met him just before he went to the US for his postdoc. Mm-hmm. We kept in close contact, so I knew a fair bit about what he was doing. And then when he came back, I don't know if he'd actually settled down to what he was going to do while I was still there. I don't think so.
Because he went to Adelaide.
Yep.
And was in Adelaide for a long time, but it was after ANU, I think.
Yeah.
Okay. Yes. So was that the step before you got to Albury to work for you?
Yeah. That was just finishing off. I was finishing off my PhD.
That's all right. So how did you get the job at Albury Wodonga Development Corporation?
Well,
how did that all happen?
Keith was largely responsible for that too.
Okay. How did that happen?
Well, the Albury Wodonga Development Corporation was trying to, I don't know, you could put it cynically and say they were defending themselves against possible criticism in the future, or more likely, I think they recognised that they were doing something that was, whilst achieving very similar outcomes, was doing work that hadn't been done before. The level of planning and all that sort of stuff was quite immense. And part of that was making sure that they didn't leave the river seriously damaged, so that the water below Albury-Wodonga should be pretty well the same as the water above.
I mean, whatever the motivation, I still, that shows full foresight.
Oh, it was, yeah. It was a time of original sort of and exciting thinking, I think, in terms of all of the government right up to the top.
And was it only you? Was it only you that?
Yes. Originally it was a consultancy, and the consultants were supervised by a committee which included Keith when he got back to Australia,
right,
and also had Bill Williams on it, and I think no, I'm not sure, I can't say who else was there. So Keith more or less sort of slipped me into to a job there, which had been carried out for a year by Roger Croom.
Okay.
Remember Roger?
Yeah. Yes, Roger Croom, who ended up going down to Tassie and then coming back again.
He did, yeah.
Was he was originally from up this area around Albury?
No, he's originally from Tassie.
Oh, okay, so he's Tasmanian.
Yeah.
Okay. Yeah. Right. Okay. Oh, there's links everywhere.
Yeah.
So you moved to Albury.
Yeah. Well, that was a one year appointment, basically.
And like 40 years later or something, you're still here.
Yes. I lost the keys to the car.
And so what was Albury like back in the early or mid 70s? Was it? It must have been a very different place.
It was 1974.
Yeah.
It was, but it was very exciting, and there was quite a number of people who were opposed to the development.
So just tell us a little bit about that development, because a lot of people won't know about what was going on in those days. That was Gough Whitlam's, wasn't it?
And Gough Whitlam and Sticky Barnard to some extent, who came from Tasmania, and another guy who was probably the most committed to the whole thing, but I can't remember his name. And they had responded, I suppose, to the view that we needed to expand the non-urban population of Australia, but that development needed to be done with foresight and planning rather than just throwing money at it and letting people come running after the money and do it that way. So, there were whole lots of issues that needed sorting out, not the least being that the Commonwealth government is not allowed to own land unless it's for defence purposes or something else. So there wasn't any way for the Commonwealth to buy up land and then organise its reletting to the community, which was basically what the job was about. So the Development Corporation, which sort of had a Commonwealth head to it, was in fact two development corporations, one for New South Wales, one for Victoria. And it functioned that way. They each owning their states' bits of land in the whole operation, which was simple enough. They had the capacity to forcibly acquire land. I don't think they ever did that anywhere, but they had a routine whereby they had the land valued by 2 or 3 people, and then they added 10% on top for their offering. So, so you, as a landowner, you'd be pretty well assured that
you get a reasonable price.
That you get a reasonable price.
And was Albury Wodonga the only place in Australia where this was happening?
No, there were others. There were a couple of state ones which didn't go very far. I think one around Bathurst Orange, and there was one in South Australia which I can't remember its name now.
So Albury Wodonga is the only one really that took off.
Albury Wodonga was the only one that had the federal government part of the framework on top of it. So the others were state ones.
So the Albury Wodonga Development Corporation building where I first started working when I came to Albury, that was the building that was built at that time? Was that? It
was built, finished in 1980, I think.
Right, okay.
Before that, we were in an army hut out at Bonegilla.
Okay.
It was fantastic. You couldn't, for instance, you couldn't use the light microscope or anything that had created vibrations or any scales for weighing things.
Why not?
Well, you could, but everyone had to stand still for the building while you did it.
And how long were you in there for under those conditions?
Oh, for 5 years.
Wow, and it must have been hot in summer and very cold in winter there too.
Yeah, well, that was the other thing you couldn't have was the cooler going, of course.
Of course.
Shook all your things to pieces.
But it was, yeah.
It had some blessings, like you never had to mix reagents. You just put things in a bottle and put them on the bench for a while.
And you were close to Lake Hume, and you were pretty close to the river too?
Yes, well, you hardly went near the town part because there wasn't any need to. And we were in building, there are two other organisations shared the building. One was the education department of the army, or that part of the army, which was actually a really good thing. It happened to be manned by people who were really committed to their job, and there were quite a few, surprisingly, more or less illiterate people who got into the army, and so they were teaching everything from physics and geography to alphabet, basically.
Wow.
Yeah. Nice guys they were too. But slightly mad.
So you were in there for about 5 years, and then they built which the building still stands on the CSU campus.
Yeah, that big one. Yes.
Yeah.
That came just a bit later, but round about that time. Yeah.
Okay.
And that was their total office.
Yeah.
They had an overall program where they were going to build head offices in strategic places, and then sell them or get them rented or something, so that would stimulate building there, and they would then go and shift themselves and build in another place.
And that was located out at Thurgoona because it was outside the Albury City Council, or whatever you call it. Is that right? Is that its location? Because even now it's 10 minutes drive, obviously, outside Albury, or from the centre of Albury, although it's now getting close to being the centre of the population. But
yes, it is. It certainly will be.
Yeah.
But you're right. There was a lot of trouble between the Albury Council, who owned a lot of the land, and the Development Corporation, I don't know how much I should say in case I offend somebody with a big stick. But so some people in the council owned land that they wanted to sell, but the Development Corporation didn't want to build there for a number of reasons, perhaps just to get even with them for buying up land and then trying to sell it back to them.
So probably considering the topic of this interview, I diverted you onto all the sort of politics and various other aspects of it. But let's get back to the sort of aquatic side of things. So, what were the sort of things that you were doing when you first got here in terms of aquatic side of science?
When I was there more or less on my own, actually Roger Croom stayed there for a year, so or 3/4 of a year or something. So he was always doing stuff.
Mm-hmm.
But his particular interest was algae, of course. So algae and eutrophication things.
And he went on to do a PhD with Peter Tyler, I think.
He did, yes.
In Tasmania, yeah.
Yeah, yep. Or did he do it in Adelaide?
Oh, look, I don't know. I just know when I was down in Tasmania doing my master's, I think he was down there. I think. My memory is probably is not so good, but I know that I had some interactions with him, perhaps, or at that time, because in Peter Tyler's lab. But yeah, I'm sure that was the case. And that was like the mid-80s at that stage.
Yeah, that'd be right. Yeah. Okay.
Anyway, carry on. So
okay. So we were given a pretty free hand, really. We had to come up with a report at the end of each time period, addressing the issue of the effects of Formula Wodonga or development in general on the river system. So we basically decided on the things we'd report on and tried to report on them. It was the standard of knowledge of the system at that point was really very poor.
Mm-hmm.
So the simple things like the fact that there are in several groups of animals, particularly arthropods, there's often a species that specialise in living in the running water of the river, and similar looking species, but often quite genetically different, that specialises in the fresh water.
In still water.
In still water,
yeah.
Thank you.
Yeah.
So, but that simply wasn't recognised at the time.
Okay.
The first time it was recognised was the craze at the time was bioaccumulation. So you measured every sort of pollutant by how it accumulated in the food chain. And so Roger, I don't want to blame Roger because it was a simple thing, but basically he went and collected a whole lot of, I think, floodplain specialist mussels, big mussels.
Velacinio.
Velacinio, indeed, yeah. And so we were going to use them all as bioaccumulators. So we stuck them in a metal-free cage for a year in the river. And the ones on the floodplain lived all right, but all the river ones died. And it was simply because they just couldn't live in this situation of flowing water. Whereas the floodplain specialists could live without water for several years anyway.
But the riverine ones couldn't.
Riverine ones couldn't.
So that was one of the first sort of recognition of these different species that had flowing water versus still water requirements.
Yeah. Yep.
Interesting. And the same thing sort of eventually for crayfish.
Yep.
The yabbies versus the Murray crays and.
Yep. And there's prawns.
Yep. Yep.
They were thought to be different. They were thought to be the same, but they turned out to be totally different.
Ah, interesting.
Yeah.
Okay.
And in every case, you can see that it's all driven by adaptations to flow or still water. But it's not only that, of course. I mean, with the still water, there's also often low oxygen. And
yes. Yes.
all sorts of other things. So,
And a lot of our, as you say, a lot of our still waters, like the billabongs, quite a lot of those are temporary,
yes,
and
indeed.
therefore the organisms that live within there have to cope with a period of drying every year.
Yep, yep, they sure do.
And so, where were those mussels collected? Were they from around Albury, Wodonga
Yeah,
area?
yep. That's something that's changed over
It
the
has,
period.
hasn't it? Yeah.
Yeah. Because we decided, or I decided that I'd do some mark recapture experiments with Eleutheria,
The riverine
the
form.
riverine form. Yep.
Yep.
And so we discovered that you could sandpaper the one side of the mussel's outside shell
Mm-hmm.
and write a number on it in felt tip pen, and then coat it over with celastic,
Mm-hmm. Mm-hmm.
and it would stay there.
Mm-hmm.
And so we were doing this in the river below Albury, where they were pretty easy to find, and putting them back in place.
Mm-hmm.
And there were a lot of mussels there. So eventually we got 500 mussels,
Mm-hmm.
numbered them all, and stuck them back where they came from as well as we could. And marked the calendar for a return visit the next year. And I bravely said, there's a slab of beer for all of the hundreds. So 100, 200, 300, 400, 500 gets a slab of beer, thinking we'd never see any of them again. They don't move at all. So they only move when the substrate or something becomes less than suitable for them.
We've done something similar. Nicole McCasker and I have been working in the Ovens River in the last few years, and, and we stuck some numbers on mussels there. It's Eleutheria again, and we did it at 2 sites, and we came back the next year to one site and we found none.
Really?
And the other site, we found almost everything, everyone that we'd marked. We just don't understand what's going on. But we've been back a couple of times, and the only thing we can think of is that perhaps that we've got our sites mixed up or something. I don't know. But as you say, the ones that we put back in at one site, they might have moved a few meters, and that's about it.
Yes.
You don't see them. I mean, the numbers in the Ovens River are amazing. I mean, they're fantastic. I can show you 1,000 in half an hour, but not around Albury.
Oh, that's interesting.
Yeah.
A lot of things have changed around Albury that weren't measured, unfortunately. Well, things like macrophytes. The water from below the weir down past Albury and Wodonga was really packed with numbers of different species of plants. There's some around, but there's nothing like there was.
I think it's Vallisneria there. Is there something like that? And that's about it, isn't it?
Yeah, that's about it, yeah.
Maybe some Nightella or something, some other type of alga I think I see occasionally down at Marina Marina.
That's right, you do. Yep, we used to. Yeah.
But there's not much, yeah. And it looks like the Vallisneria is really quite prolific where it occurs, but not much else. Yeah. But there's been, as you say, there's been a massive changes in the system. I often talk to my students about, you're interested in climate change, go and have a look at the river there. It's been
changed.
Its climate's been changed seriously for a long time. Yeah. That's really interesting, because I do talk to my students, my river ecology students, about the different species that sort of specialise on floodplain versus the ones that are actually in the river, and that down in the lower lakes and the lower lakes and the locks, the lower Murray, that there's been basically a loss of the Murray crays down there, and also the change in the mussels. I think dominance as well. So that's interesting to see where that came from. And Keith went on to specialise or be fascinated by mussels.
He got a grant from somewhere to write a report on mussels in Australia.
He's fascinated by them. Even up until his death, he was still playing around. He told me not long before he died that he had bought with the royalties from our book, would you believe? He'd bought, I think it's Mathematica, a mathematical program. I don't know anything about it.
Oh, yes.
And he bought it primarily so that he could look at the measurements of mollusks, the various
Oh,
metrics,
right.
and then adjust those measurements, expanding some and
Yeah.
contracting others. And he said that he could construct almost any shell, whether it be a gastrolith or a bivalve, just by altering a few of those parameters.
We
Yeah.
did a lot of work on shell shapes between the large mollusks. They always overlapped, and we never got a very good grab on
it. They're a difficult group for all sorts of reasons.
They are, but fascinating. I mean, their life cycle, for instance, is just ridiculous.
Yeah. I was talking to Nicole McCasker about that because she's, it's her passion at the moment, these mussels, and we're going up to the Darling River actually shortly to do some survey stuff. And as you say, the mussels have explain to us what the life cycle of a mussel is, of our freshwater mussels, rather than me doing it.
Why shouldn't you do it?
No, because I'm interviewing you and you're not interviewing me.
I see.
That's the difference.
I would come and ask you that question. Well, if they start off as an egg, let's say. Well, first of all, the important unusual feature of their life cycle is that because their lungs are also involved in moving water around inside the shell, and indeed in keeping oxygenated water next to the tissues that transfer the oxygen to the body of the mussel. So the eggs are laid on these tissues, and I can't remember what the name of it is.
Gills?
Gills will do nicely, yeah.
Yeah. Yep.
But they're more than gills because
they're lamellar. Not lamellar? I can't remember the name of it to myself, but it's like a W or M shape, depending on which way you look at it. So gills.
Yeah.
Lamellibranch. Lamellibranch gills.
That's right. Yes, that's exactly.
I don't know. I'm not quite sure what lamellibranch actually means, but anyway.
No.
But that's what they are.
Yeah. So eventually the eggs, they hatch inside the shell around the lamellibranch, and released into the water, probably often stimulated by the outside conditions in for the adult mussel. So for instance, a fish may nudge them or something, and they release these things wherever they can, because the next step of the life cycle is for these little tachas. They're not called tachas, they're called something else.
Placidia.
Placidia, thank you. Are released into the water, and they're like little hamburger buns that bite.
Yes.
So they tend to be able to snap, and particularly when they're moved, and more particularly when they're near a fish or other aquatic creatures, they'll attach to a few. They catch on and actually stay there for about a fortnight, during which time it used to be thought they were being parasitic on the fish or other creature, let's just say fish. But that's probably not true. They're just hitching a ride.
Just hitching a ride, yeah.
But the fish builds a cyst over the top in much the way as if you leave a splinter in your finger, eventually you'll get a little blister over the top of it, and then it will go away. Then it will break and the insides get out. The same thing happens to these little guys. So the adult mussel, nothing. The baby mussel grows into something that looks a bit more like a real mussel than a hamburger bun, and is free to roam the world, sticking to the wet bits.
So what's your thought? And we were talking about this the other day. What's your thoughts about I mean, not all freshwater mussels do this, do
No,
they?
no.
But obviously, the hyraeans, the family, I think
Yeah.
that we're talking about, they do that. And you say that at one stage it was sort of to be a parasitic sort of thing, but you think they're just hitching a ride.
I think so. Yeah.
I mean, probably that's still a bit contentious.
Yes, perhaps so.
But why would they do that? Why hitch a ride?
Well, for dispersal reasons, we've already seen that at least in some cases the mussels are not exactly wanderers,
Yes.
and like most creatures that produce a whole lot of young, the individuals are fairly dispensable. So um, the trick, the obvious trick to keep the population going is to be spreading wide and
Yeah, but if I was devil's advocate, I'd say like there's a lot of fish, especially in flowing parts of rivers, that will just broadcast spawn their eggs into the water and let the currents do that, not
yes,
having to associate with fish or
yes, that's
other
right.
fish.
Yeah, that's
I can
right.
see in stillwater habitats, in
Yeah.
Atlantic
Mm.
habitats, I can imagine that dispersing would be more difficult for
Yeah, yes,
something
you're
like
right.
that. But I don't know.
No, makes sense. I don't know in that case.
But look, I agree. They're a fascinating life history. Absolutely fascinating life history. We were just looking at some the other day, and of the Glaucidia that snapped onto the lips of the larvae of Murray cod, which was really interesting.
Ah, well, they I don't know if they favour the youngest of developing forms of fish and yabbies and things that they get onto, but we felt that they were less successful on more mature fish.
Ah, interesting.
But we didn't publish it because we weren't actually working on it. We were interested to follow the life cycle,
Yeah.
which
Okay.
is very hard because when they leave the host, they're still very small. And it's hard to find them.
Yeah. Yeah.
But of course, being small like that allows you to get into the substrate a bit more easily.
Yes, and at that stage you're just looking like a little grain of sand, or not much bigger than
Yeah,
that. So,
yeah.
yeah, no, I can imagine that. Um, so you were, if we sort of get back to what you were doing in Albury, I mean, you've been talking about that anyway. Um, I was clearly the federal government, Commonwealth, was interested in monitoring the effects of what was going on, development in the river. But and you sort of said that as long you provided reports at the end of years about what was going on. How much freedom did you have personally in sort of working out and determining what sort of research and monitoring you did? Was that sort of just decided by a committee, or?
Yeah, it was certainly looked at by that committee that Keith was running at the time. And after I came there and we set up a more permanent arrangement, that supervision got less and the input from the planners and the corporation got more. But basically, they just wanted reassurance that they could do what they had planned to do and not create terrible trouble.
And yeah.
So, for instance, when they built the paper mill here, um, we were quite involved in running monitoring programs of all sorts to see that it wasn't going to do any damage, which it probably wasn't, and it probably hasn't.
So when one monitors something, I mean, there's obviously been a lot of development of a lot of different sort of techniques and methods and protocols.
Yeah.
But generally you have to have an idea about what is, the baseline, what is the reference from which you're monitoring things moving from. Was that something that you had enough time to sort of go, well, we need to sample for several years, particular groups of organisms in parts of the river and floodplain establish that baseline, or did you have to,
I think
basically?
we were given a lot more trust in those days, and
Mm.
and the idea of the community having a say in what you do and so forth wasn't uh, particularly strong. So mostly that was a kind of open-ended thing, in that we developed a sampling program that would help us compare. Um, my brain stopped.
That's all right.
Set up a normal experimental setup, but having to say when we'd got enough information and so forth wasn't really an issue. Starting the job was enough to mean that you could finish it.
Yeah, yeah. So, but determining whether things were going up or going down or sort of going sort of flat, that must have been a difficult thing to know.
Yeah, it is. Yes, yeah, and I'm not saying that we'd get away with it nowadays.
Although it's interesting because, I mean, was that about the time when the sort of Murray monitoring with, I know that John Hawking was involved with that for like 30 plus years of putting baskets for colonizing invertebrates. Was that when that started?
No, that started almost immediately.
Okay.
I don't know if it started before I came here or soon after, but it was, and we did a bit of experimentation on what the baskets should look like and how you can try and make them as uniform as possible.
Because I mean, in some ways, it's like some of the stuff that I've been involved with in sort of making use of historical collections that weren't ever designed to provide the information that we can now extract from them. That almost irrespective of the reasons why that was started, it's a gold mine in terms of information that has been monitored for such a long time.
Yeah, it is.
That it's sort of got emergent properties from which we can draw things now that we probably never knew, never envisaged in the past.
Yeah, well, one of the driving forces was the fact that the Murray-Darling Basin Authority recognised it as something useful after it had been going for a while, and were very supportive in keeping it going. In fact, for a period, put like traps all the way down the river system. The most limiting issue, I guess, was taxonomy in those days, because no one much was doing any collecting systematically, and certainly the only people who were trying to be taxonomically correct was the Victorian Museum, who did some good work, and we eventually worked together with them, which was the beginning of the taxonomic workshops that we used to have every year.
Yeah. I'll ask about that in just a minute. But so there must have been there's obviously some perception that, or obviously idea that that the macroinvertebrates that colonize those baskets gave you an indication if they changed, that something was changing in the river.
Yes.
And that presupposes not just the fact that the organisms, the assemblage of organisms is changing, that tells you something, but that the nature of that change, that the types of organisms that might come in or the ones that you lost have some degree of preference or tolerance to the conditions either they started off with and then change, or they come in and go out. So that's to me a quite a sophisticated. I mean, in retrospect, we know all that now, but at the time, that's a fairly sophisticated way of looking at things. And if we're talking about it's probably around the time of just after the river continuum concept came
Yeah.
in, and the
Yeah.
functional feeding groups and
Yes.
the understanding about that. Was that river continuum concept something that resonated in people's minds at that time?
Well, I don't know. I thought it was largely self-evident. I must say, it didn't seem so exciting as other people saw it. But
Seems to have done very well over the years.
well, there's a certain section of academia, in particular, in the US.
I'm very excited by it.
They thrive on naming stuff, and naming it doesn't affect your understanding of it much. So,
But I mean, to be fair, apart from, I mean, there are obviously a few
don't think we should be fair on that.
but I mean, like, you know, I'm thinking about the history of sort of understanding about how rivers function, and you know, as I often tell my students, that the science is fairly young. I mean, it's been going for quite a long, you know, depends on how far you get back and what you want to talk about. But you know, Noel Hines's Running Waters book was, I think, 1970.
Yep.
And then 10 years later, with the river continuum concept coming in, it was, I suppose, those sort of concepts, and I've been guilty about coming up with a couple myself, are sort of things that people can hang their hats on. And I think people crave, scientists just as bad as anybody else, that look for something that somebody else has come up with that's given a name that they can then cite, that they can reference, that they can then also criticize. I mean, it's a hypothesis, I suppose, and that's how science works, isn't it? A model that then you can destroy or whatever, or build up as you like. I don't know.
Yeah. Yes, I guess that's true.
Gives you a view. But you don't think it wasn't something that you think influenced that work?
No, it's more likely. No. Oh, well, it's hard to say.
Yeah.
Certainly, it would have come into thinking about it and writing about it. That's for sure.
But as you say, the taxonomy workshops. I mean, when I was at the MDFRC, and I contributed to one of those,
yes,
that they were, and I was talking to, I think it was maybe Rhonda Butcher when I did the interview with her. Maybe it was after our recording had finished, but they were something really special. I mean, you know, that for all sorts of reasons, and we've got at Charles State University, I've got all of the identification guides there.
Have you?
A complete collection of them, which we use in our teaching. Yeah. And they're an incredible resource. Absolutely incredible resource.
Yeah. Well, it started. I'd done although I was working in the lab most of the time, I did a lot of other stuff for the Development Corporation, like producing models for people coming here to establish industries and those sorts of stuff. And I started a system of classification which used eight numbers or 6 numbers, I forget which, and you could tell from that everything from class down to individual with other numbers for immatures, all that sort of stuff. So it was a way of classifying. But it was a way of classifying animals that you couldn't classify. So it was just, so for instance, and that was also built on what the museum was doing at the time, which was to give letters to different names. So something species. I remember a coronwood species J. It's one of my favourite creatures. And
right.
And we knew, so we could have had a species J as well, but we'd have no way of knowing if it was the same species J or something different.
So you came up with a numbering system that was a unique one.
Yes. Mm-hmm. And so that meant that in order to make that work, we had to get together and, you show me your species J and I'll show you mine.
Mm-hmm. So a reference collection.
A reference collection is exactly what it was.
Yeah, okay.
Yeah. So that's where the interest in taxonomy started from.
Makes sense. Yeah.
and gradually moved to all sorts of other things.
Well, I mean, there are I don't know, there are 30 plus, I think, of those, maybe more. I'm not sure, of those identification guides and associated with the workshops. And you look at the names of the people who are involved with those, and they're luminaries in our field.
Yeah, well, that was the good part of it. People just cooperated. They seemed to want to do it.
And they did it for free, I think, didn't they?
Oh yeah. They certainly didn't have any money to pay them.
And they came every year and gave these workshops and developed these keys, and all the strange people who were interested in these animals came out of the woodwork to attend.
Dragging their knuckles.
Yes, I remember going to some of those and thinking, well, I didn't even know these sort of people existed. But that was, it really contributed enormously, because it was the rise of the understanding, like I sort of mentioned before, about the preference and tolerances, the recognition of these preferences and tolerances of especially the arthropods and insects, and the development of things like Oz rivers and the different water quality indices and things.
Yep.
Using those. What was the riv packs, all that sort of stuff.
Yeah.
So that would have been invaluable for that. But a lot of those ones were originally developed for sort of water quality rather than quantity.
There was a strong belief in those days that you didn't have to do any chemistry. You could look at the biota, and once you knew enough about them, it would tell you all the chemistry you needed to know, which hasn't probably proven to be true. It was very unlikely to be true in the beginning.
But there are a lot of advantages in using those animals, because they do integrate over time. They can tell you.
Yes, that's true.
I remember going to a river some years ago, and we went in, got in there, and sort of dipped into the round electrofished as well, and found nothing, absolutely nothing. The water quality was beautiful. It was, the pH was 7 or something, and the temperature was great, and there was low conductivity. There was all sorts of wonderful things in there, but almost nothing. We couldn't work out why there were no animals in there at all, until we went and talked to the publican, who told us that there was no water in there about a week ago. So lack of animals tells you that there's been something happening there.
Yeah, I think if I came upon complete lack of animals, I'd go and check my crocodilometer.
Well, there were a few bits and pieces of humans lying around, but apart from that, we've been talking, would you believe, for an hour and 5 minutes already. I know, and we've only got sort of the first, I think, 20 years of your career so far.
Oh, that was the best fun.
So we might have to reconvene at another time to get the next 20 years. But just to maybe we just sort of wrap up this particular interview, if that's all right. So you were the Murray-Darling. I'd just like to know where the Murray-Darling Freshwater Research Centre actually came, because at the time.
Oh, it's complicated.
Oh, it's complicated. Oh, it's going to take too long, is it?
No, I'm happy. I'm not doing anything else.
Maybe can you just explain from? I mean, originally the Albury Wodonga Development Corporation and the work you were doing with CSIRO, is that right? Predated the Murray-Darling Freshwater Research Centre. Is that right?
The Murray-Darling Freshwater Research Centre started, I think, in 1981, but it could be a year before or after. I'm not sure. The reason it started was a lot of pressure from a South Australian federal politician, and his name escapes me at the moment, which is a real shame, because he was a great guy. Anyway, I think I might have one of his books somewhere. Anyway, he was very keen that there should be a freshwater research effort somewhere in Australia. It should be, and it should be based on research, but also applied research.
Because you said CSIRO wasn't all that keen up until
No,
that stage.
and they weren't right until that stage. Of course, and the sound of money probably made a bit of a difference. But anyway, so eventually there was he put forward a private member's bill, and the result of that was that that I'm sorry, my memory's going out now. That they had a Senate committee looked into it, and I think I'm right in saying that was headed by forget. So their conclusion, largely managed by a really canny public servant, whose name escapes me now, who's kind of was pulling all the strings in the right directions, that they would set up a research centre, that it wouldn't start a new one, they'd base it on MDFRC, and that they'd invite CSIRO to manage it. So that, with a few minor changes, was the beginning of the MDFRC.
Okay. Which until fairly recent times maintained that name. So.
Yeah, it did, yeah. And it was, so David Mitchell was its first director, because part of the deal was that CSIRO should provide the impetus for research and so on.
Yeah. Okay. So David, I think, had been up at Griffith.
He had. He was the chair. He was the director, chief of irrigation.
Yeah.
Irrigation research.
He'd come from Zimbabwe, or it was still Rhodesia in those days. I'm not sure.
It was.
I'm not sure whether it changed. Yes, and he was director for many years before you took over.
Yep.
We might stop there for the moment. I think we'll. I'd love to keep going at some other date, if that's okay. Because I mean, I haven't really tapped into your views about all sorts of things, about how things may have sort of changed in terms of the focus of research and the approach to research and management too, which I'd like to talk about, because I've, in my 25 years or so up here, I've seen a lot of changes, but you've seen a lot more than I have, and I think giving people, especially the younger generation, an idea about, like I was asking about the autonomy that you had and who made decisions is really interesting, sort of how things have changed and how much regulation of what goes on compared back then, and where the money came from and things. But anyway, we can talk about that at another day.
Yeah, no,
that's okay.
I'd like to do that.
All right. Well, that'd be great. So thanks very much, Terry, for giving up your time on this Friday afternoon, and it's almost beer o'clock, I think.
It is.
Well, it's really past it.
Even now, after 20 years, I still occasionally stagger out on Friday and look around.
Nice. In these strange COVID-19 days.
Yes.
And I asked Terry before I started recording what the picture of was behind him, and it's the Darling River, you were saying?
Yeah. About 20 or 30 kilometres upstream from Wilcannia.
And why have you chosen that picture to be your backdrop?
I like it. It's one I took myself, and it was just after the drought broke in, whenever the
2010 drought break. Is that
no,
the one? No, much more before that.
Yeah, back in the end of the 70s, beginning of the 80s.
Okay, because there was
beginning of the 80s,
okay,
82, 83,
mm-hmm.
And there were all sorts of exciting things happening, like the Darlings getting stirred up and so forth, and getting bogged and all those things. And this is a really, not functional anymore. In fact, you can see the posts. If I got out of the way, you can see
Oh yeah, yes.
posts going up there, which would have carried the pipes for the pump, and the big wheel up the top there would have taken the pump to the steam engine that was driving it. So it was a very early irrigation pump along the Darling.
How old do you think that pump would be?
It would have had to have been in the first quarter of the 20th century, I reckon.
And what were you doing up there at that time?
Oh, I was chairing. There was a fad for having expert panels look at particular rivers. I guess it's died by now, but I was.
Oh, I think they still occur.
Do they? I was doing one on the Darling, but it turned out to be good fun, but weird.
Well, I think, look, let's leave it at that. On that note, of the weird expert panel on the Darling River, maybe we'll reconvene and I'll ask you what was weird about that panel. Maybe the people that were on it, maybe the things that you were doing. Anyway, but thank you very much for giving up your time, and we'll talk again, and then hopefully in some ways better circumstances, because we're sort of still under the thrall of this strange virus that's hit. We'll look back on this time, I think, with all sorts of wonder and surprise that we were so surprised by it. Anyway, thanks again, and we'll talk again. Thanks, Terry.
Thank you. And I'm sorry I was late.